So I think it's time to slowly start. So hello everyone, I welcome you to another session, and it is my pleasure to introduce Hsiang-Yun Wu, who is postdoctoral research fellow at the Institute of Visual Computing and Human-Centered Technology at TU Wien in Austria. Yun received her PhD from the University of Tokyo, Japan in 2013. And I have to say that Yun is an amazing scientist with interest in different visualization fields. However, she is the best known within the community for her work on visualization of complex networks and biological pathways and road networks. So I'm very looking forward to her talk about this topic. Yun, floor is yours.
So thank you very much for the nice introduction. And yeah, so today I will be talking about biological network visualization. My name is Hsiang-Yun Wu, and usually I ask people to call me Yun, so it's easier to communicate with people with this name. So today I will probably show you a bit quite different image from what you have seen within this week. So it will be basically about graph structure and how they are used to visualize and show the complex relationship. So the graphs are actually used everywhere. For example, you can see here, it's used to represent biological network where each node represents a kind of chemical component, and the relationship shows the connectivity between those chemical components. And in the brain network, usually shows the functional connectivity between the brain region via statistical analysis. And we can also use network to represent blood vessel, where how human blood is flowed through the vessel. We can also use it for representing food web, how those people are eaten by the other animal, and how the supply chain are consumed. And we can also use it for representing ecological network. In this, for this reason, you see that basically network exists for nanoscale and also appear in gigascales. The size of network is also diverging a very wide range. For example, you can see this very tiny graph which only contains a few nodes, as you can see with this image, or you can have a larger graph contains about mesoscale record, or you can have even larger one, more than 1 billion, you can have a macro scale relationship description. So there's only one thing we can guarantee: as long as the size increase, practically, if you want to visualize such a graph, you usually get this kind of lovely hairball effect. So why we are using graph? As I explained previously, this is an intuitive representation for visualizing a relationship. And a graph drawing is actually a scientific field that we investigate a pictorial representation of a graph. It could be node-link diagram, as you have seen in the previous, and here on the upper right, that vertices and nodes are usually represent as circles and labels, and edge and links are usually represents as lines or curve or arrows in the Euclidean space, as you can see here. But besides node-link diagram, you can also use matrix representation to represent the relationship. For example, now this node 1 is connected to node 4, which is, and those are the basic concept of graph. And what is the difference between network visualization and graph drawing? As I mentioned, graph is practically a representation, node-link diagram is practically a representation of vertices and edges. And for network visualization, it is often associated with attributes and other additional data. If you remember the talk from Alex yesterday about the multivariate graph, that is the case. So practically, why drawing a node and an edge, or a set of nodes and a set of edges, is a difficult problem? If you consider the most popular hand draw map in the world. I would say it's a London Underground subway map. In order to improve the readability of such map, the designer actually tried to simplify the trajectory of the paths to make them align to certain orientation in order to improve the effectiveness of tracking a single or multiple path. And there are several design criteria has been incorporated in this diagram. And having all this kind of design criteria at once, which will generate a very complex optimization problem. And besides, this is a semi-automatic approach where we have the world map of receipt, where each node represents either the ingredient of food or the name of a receipt. And as you can see here, compared the quality in the previous image is not as good as you would like to have, but with the additional add-on, it's still acceptable. So today I would like to talk about what are the current state of the art of such an automatic approach can assist human designers, and what are still remaining to be solved in the future. There are also other interesting diagrams. If you are interested, you can take a look from those lists I have collected. So practically, graph visualization can be categorized into two categories. One is with spatial data, such as metro map, as I showed before. Another one is non-spatial data, such as biological pathway. And you can see here on the left bottom corner is a cancer pathway representation using subway style. However, creating such a diagram is not a very intuitive process because all the designers have different design preference, and also the target group have their own expectation. And it doesn't mean that all machine, even a machine accept all those criteria will generate a good or visually plausible result. So usually this is a bi-directional process where we start from the human perspective and then pass to the design perspective, and the machine try to solve the question, and then you are doing the bi-iterative loop in order to find out what is the best outcome out of it. So biological network is actually any kind of network that applies to biological system. So according to the literature, usually it has this small word property, which means that they are highly clustered and with a lot of shortcuts that connect to the clusters, which made it especially difficult to draw because you will always have these high degree nodes, which is actually the origin of causing this kind of halo effect. And besides, there are several types of biological network, for example, protein-protein interaction network that describe the interaction between proteins, and this sequence similarity network that shows the similarity between genome, or other many. And for example, like metabolic network describe the reaction within of human metabolism or other metabolism in life form. Besides those biological network, there are also additional one. For example, like ecological network or epidemiological network and species interaction network, food web. They are also very different scales of network. And why this is challenge? Because of the increase of data. As you can see, CAKE actually is one of the most popular pathway database. Then since last year, they have over 700,000 reactions recorded in the database. And this data is usually heterogeneous and evolve over time. And the knowledge is even more, most of the time is domain specific and domain dependent. And the increase of data usually increase faster than the computational scalability and perceptual scalability. So without a good visual management strategy, it's very effective efficient algorithm. It's very difficult to understand what's the relationship hidden in those graphs. So let me show you a diagram how this graph should looks like. I hope you don't have this typophobia disease, so you feel a bit uncomfortable with this diagram. So this is very dense with this limit of screen space. Maybe you have seen this diagram from Young's presentation on Monday, but this is the new diagram they released last year. So because of this difficulty, we always need to conduct
So I think we have some technical issues, but in the meantime, I can at least test my audio. Is it better now?
Yes, Jan, it's better.
Perfect, thanks. So let's see if we can get Yun back.
Yeah, maybe I can keep you company while we wait. It's so awkward. It seems to be she's having some internet issues, so I hope we can bring her back.
Oh my goodness. All of a sudden she just dropped out.
Yeah.
I'm sorry, I think I got an error and got kicked out.
No worries, welcome back.
Oh, sorry, I was not aware of it. Where am I lost?
So you were explaining this large chart.
Okay. Okay, so I will start with. So this one, right?
Yep.
Okay, so yeah, so practically I just want to show how complex this diagram is. Okay. So this unfortunately lead us to the upper corner of this data complexity matrix that we need to perform visual analytics for analyze such a big data. So this is an overview of existing approach. There are many network visualization approach, but what I wanted to say here on the left-hand side, those are more oriented machine-generated diagram, and on the right-hand side, there are handcrafted diagram. And as you can evaluate from the visual quality, on the right-hand side, you will have higher trust and better visual quality. So this is the classical visualization pipeline. I think I can skip this for the moment. But what you need to do in advance is to understand the task that a user or the domain expert want to perform. So usually in network analytics, you need to perform topology tasks where you try to find the neighbor connectivity and also perform the attribute test and also the browsing test to find the path and also try to recognize the functional groups in the data. So the problem is, even though we know there are nice connectivity between metabolites and also cool reaction connected together, the problem is, oh, I cannot find what I want to find in this large diagram. So unfortunately, this kind of hand-craved diagram was crafted with 5 undergraduate students within 20 months, and they have to manually create this type of diagram, which is very time consuming. And our idea is to how to improve such a complex process. So in this visualization pipeline, I can further divide it into data form and visual form and interactive form. So in the data form, it is interesting that in biological pathway or biological network world, there are many representation for network. So you can have a single simple graph, or you can use geometric matrix to represent the relationship, or you can use a substrate graph that each node represent a substrate, or use a k-parta graph where you introduce additional reaction node in the graph, or use a hypergraph to represent this type of data. In addition, you can also represent your graph as reaction graph, or you can have the cluster information embedded to have cluster graph. Nowadays, it's more common to have a more high-level concept called a multi-layer graph, where you can embed all the previous graph model in a whole, so that it's easier to conduct analysis. The problem is, can we have an effective algorithm to access those data or those relationships, or can we have an effective multi-level graph visualization? Yes, so for the visual form, it's very complicated. If you see, probably you see nothing on this diagram, but you still can somehow recognize there's a circle here, there's somehow chain over on the right-hand side in this zoom focus area, or you can see some chain at the bottom corner. So there are classical aesthetic measure that you can incorporate the network visualization to align the edges and nodes in order to achieve certain quality. But if you want to add more attribute, for example, even for the name inside this large diagram, or some chemical representation in this diagram, it is very difficult. But for the biologists, they would like to see the detailed representation as well as the high-level functional groups to simultaneously with network visualization. So conventional approach is, okay, if we try to dump it to a so-called force-directed layout algorithm, we will see such nice complex result that we don't understand anything. But if we try to organizing in a way that, like by incorporating the concept of urban block, as you can see here, try to recite the chemical reaction in its proper area. As you can see here, they can be more effectively arranged. And this, as also you can see previously, most of the designer prefer to use orthogonal representation, which is not a very scalable algorithm. Usually, I think until 500 nodes, it's probably the optimum. But with this kind of divide and conquer approach, you can somehow manage to scale this kind of orthogonal approach to a certain degree. So this is done by incorporating many aesthetic criteria, as you can see here, by aligning similar functional group together with each other and try to overlap between each other and try to keep the pairwise relative positioning. So this is done with a formulation with a set of a linear constraints so that we can solve it with linear optimization. So here you see the quick result. I would like to just quickly go through how this is done. But the interesting part here is we try to create flow networks to guide the edges to avoid also intersection between the nodes and edges between the city blocks and within the city blocks. So as you can see here, we managed to scale this kind of orthogonal representation into a certain degree, and with the similarity box alignment, we managed to have those similar blocks automatically aggregate together. Actually, we discussed with the designer from Cake, but we didn't talk about how they assign the color here. But apparently, it's a proof of concept that we found out that they actually try to put those with similar or those shares the similar metabolite together with each other. And this is another result with the same diagram, as you can see here. So with this approach, we managed to somehow save the designer's time from 20 months to 2 hours by summarize a lot of motif and multi-scale representation, as well as interactions. Yes, so what about, I mean, if you take a look at the classical representation created by human, it doesn't have this kind of artificial rectangular representation, which is kind of artifact from the previous approach. So our next approach was trying to find a more flexible boundary representation for such type of complex network. So what we did is we tried to incorporate the multi-level concept, where we try to roughly decompose the screen space into certain region, and based on different level of detail, we try to embed the network within certain region and try to keep balanced distance between each pair of vertices. And this has been done again by a representation, by incorporating several representation, and the boundary optimization is done by introducing the concept like metro map representation. So we constrain the boundary into octilinear orientation, like 0 degree, 45 degree, and 90 degree. So with this, we can actually create such a bit more generic border diagram, where you can see the graph are aligned within their own region, but still have a balanced distribution, as you can see here. And with the approach, we can also create the layout with different aspect ratio. So this is again the world map of receipt, as you can see here. And this is the same Cake data set, where you can see that the vertices are reside at their assigned region. So our idea is that, okay, well, how far we are still from human design diagram, as Laura was presenting in the previous talk, that she was trying to create a biological illustration. And for us, it's like, okay, how can we identify whether this graph was drawn by the human or by a machine? So we conduct a Turing test to have a pair comparison between the hand-drawn network and also the the machine generated diagram. And we realized, well, most of the time human can still recognize what is drawn by the machine, especially for those orthogonal representation or circular representation has a strong tendency. But for approaches like spring force algorithm, actually, or organic layout, actually most of the human cannot already cannot identify what's the difference between machine-generated and human-generated layout. And in the interactive form, I would like to briefly talk about there are several interactions can be added in the network visualization. For example, like select the subset of the network, or explore the network, or reconfigure the position of the network, or try to encode different visual encoding in the network, or try to abstract or elaborate some certain sets, or try to filter out what is important to be visualized, and etc. So unfortunately, I have to say, so far there are not many good, I wouldn't say not good interaction approaches, but they have common disadvantage that they are very constrained to the overview diagram. So usually if you visit the classical database, you will see that they usually offer you a overview diagram, and based on this diagram, you can zoom in and zoom out to see more detail. However, because those even those small diagram is semi-automatic created by human. What I mean by semi-automatic here is that you need to use tool to drag around the vertices and edges in order to have certain good layout representation. And yes, the same for the cake. For the cake database, you can practically select the nodes and check the attribute detail, or see the detail of the chemical structure. But to be honest, if you don't remember all the relative position on this diagram, you have to hover all the individual vertices in order to better find your target, or you will need to try to search using a tool in order to highlight what's target to be. Where is your target vertex or edge or reactions? And so far, Reactom is the first approach that I know that the layout is fully automatic, but only until certain degree. So they have this tree-like structure to visualize the ontology of the pathway. But as long as you go or dive into the detailed representation, the final fine pathway layout is still drawn by the human. Yeah, maybe I can show this. Should be some, yeah. So for example, such a layout. So yeah, I will still want to have some time for questions. So I would like to wrap up here. There are several challenges still remain for biological network visualization. For example, there are many standards for data acquisition and how we can fuse this knowledge together and manage it. For example, there are graph database that allows us to do the crowdsourcing for collecting the relationship of in the pathway. But how can we guarantee the quality and the trustworthiness of the input data? For example, like the recently published COVID knowledge graph, the author mentioned that at the beginning, the data collect was very fuzzy, and most of them are from the social media. However, as long as the time proceeds, they try to improve by investigating a lot of human resource on checking the published paper and the literature. So, yes, so it's still a challenge. As you can see from my presentation, there are different overview maps for different databases. So it's still even a challenge to relate yourself. They are representing similar stuff, but some of the database they prefer to use undirected graph to visualize the network, but some but some database prefer to use directly graph to represent the network just because they are having a different view on the significance of the age direction. And these uncertain patterns and visual representation confuse a lot of research, where if you are used to one representation, and it's very hard to move on to another one. And also finding this kind of pattern embedded in the network is still a very challenging task. For example, what are topological pattern matter? What do they have biological meaning? And what visual patterns matter, for example? And we are still lacking effective and scalable layout algorithm. And hopefully in the future, we can also have multi-level and scalable interaction, not just simple pan and zooming approaches. And even it is difficult to have usability testing in large network scale. So I would like to thank my collaborators. So with this, I would like to finish my talk. Thank you, and sorry for the technical issue I have in between.
Thank you, and no worries about the technicalities. It was a great talk, nevertheless. There are actually some questions, and I think we have a little bit of time to answer them. So Michael is asking, where did the increase in the number of nodes and edges come from in the metabolic techniques, whether it is cloning of currency metabolites or other strategies?
Sorry, I have a delay. Could you repeat it again?
Maybe I post it into the chat. Is it
yes, that would be great. I will need to, if I can stop sharing right now and can see the chat.
So can you see the message?
Yes, I'm reading it right now.
Oh, yeah,
The increase of number. So for the metabolic techniques, there's no increase of nodes and ages. Practically, the data come from the existing RECOM map data set or the existing CAG data set. Now I know. Okay, so there's a I didn't mention it at the beginning. For the biological pathway, it's very difficult to visualize the whole graph in a whole, because, for example, there's usually this bad guy water molecule which practically connect to all the chemical reaction in human metabolism. If you have one node that connects almost 80% of the other nodes, practically you cannot see it. For this type of nodes, we have discussed with our domain expert, and practically they consider those are unimportant vertices. So for those unimportant vertices, we try to duplicate the nodes to improve the visual, to somehow improve the visual clutter from the existing network.
Thank you. And the second question, I'm posting it again into the chat, but for the audience, Laura Garrison is asking, how do you think about the balance between simplifying the pathways down to that they are more comprehensible? Do you think there always needs to be a human in the loop to identify key or hero molecules in the signaling cascade?
I do think so, because the problem is for those kind of simplification approach, you cannot really assign biological meanings to this simplified structure. That's why most of the domain expert would really prefer to have a very detailed network printing in a very large scale so that they can know the detailed connectivity. But it's very hard to say what does this mean if there's a water molecule connected to a high-level functional group, and how can you describe it in a biological way? It's difficult. Yes, and I would say right now, the machine generate approach is still not very good enough to really achieve all the aesthetic criteria designed by the user or the domain expert. So I think right now it's still important to involve user in the loop, and hopefully in the future, if we can develop more scalable approach that really can generate high quality result, which that's why I'm still not stopping and still working on this topic, then maybe in the future we will have this as a chance.
Thank you very much for both the answers, and again, really great talk.